Hello friends, today I am going to talk about aquaculture practice of finfishes, crustaceans and molluscs. Let us commence with a definition. Aquaculture, also known as aquafarming, is the farming of aquatic organisms such as fish, crustaceans, molluscs and aquatic plants. Farming implies some form of intervention in the rearing process to enhance production, such as regular stocking, feeding, protection from predators, etc. Farm raised fish, for instance, grow faster than wild fish. Aquaculture involves cultivating freshwater and saltwater populations under controlled conditions and can be contrasted with commercial fishing, which is the harvesting of wild fish. Broadly speaking, finfish and shellfish fisheries can be conceptualized as akin to hunting and gathering, while aquaculture is akin to agriculture. Mariculture refers to aquaculture practice in marine environments and in underwater habitats, but there are disadvantages too. An aquaculture business requires large initial investment and diligent upkeep. Fish grown in captivity catch and spread bacterial infections quickly, and they can easily deplete the oxygen supplies in a pond or cage. Let us know something about culture of finfishes. First is Indian major carps. Amongst the group of cyprinids known as the Indian carps, three species are of greatest economic importance. They are Katla, Rohu known as Labeo rohita and Mrigal, Sirinus mrigala. Under farm conditions, the Indian major carps grow rapidly, reaching marketable size within 1 year in a tropical and subtropical countries. The Katla is the fastest growing species. In natural waters, it frequently attains 300 to 400 gram in the first year, over 2 kg at the end of the second year and 5 to 6 kg after 3 years. In fish farms, however, they can reach more than 1 kg during the first year. Rohu do not grow as rapidly as Katla, attaining around 500 to 1000 gram in the first year under good farm conditions. The growth of Mrigal is poorer than that of Katla and Rohu. As is the case for all cultured fish species, growth of farmed Indian major carps is a function of stocking density, availability of natural and supplementary food, competition with other species in the same pond and environmental conditions. The food and feeding habits differ between three Indian major carp species. Katla are column feeders, mainly collecting the zooplanktons at all depths. They occasionally also feed on the bottom, utilizing organic detritus, remnants of aquatic plants and mud rich in organic matter. Thus, when stocked together, Katla and bighead carp can be competitors for the natural food supply. Rohu is a mainly bottom and column feeder, preferring plant material including decaying vegetation. The Indian carps do not breed naturally in ponds or other confined waters. They are all river spawners, having semi-floating, non-adhesive eggs. The eggs have a large perivitelline space and during swelling, they increase their size by about 10 times. Swollen, water-hardened eggs are rather delicate and do not tolerate physical shocks well. This sensitivity to mechanical damage is a very important consideration when Indian carp's eggs are to be incubated artificially. The Indian major carps normally attain sexual maturity in their second year. However, as with other cyprinids, age at a sexual maturity is temperature dependent. It can be delayed by 1 to 2 years in the northern extremes of the fisher's range. The fecundity and breeding habits are similar in all the Indian major carps and they can be considered collectively. Though fecundity naturally depends on the nutrition and living conditions of each individual, generally females spawning for the second and third time produce relatively the most ova, that is 1 to 2 lakhs eggs per 1 kg of body weight. In nature, spawning coincides with the onset of the rainy season. Under natural conditions, the Indian major carps spawn in groups once a year. Thus, at any one time, all the eggs in the ovary are in approximately the same stage of development. In cultured conditions, they can be induced to breed twice within the same spawning season, the second occasion following about 2 months after the first. After natural spawning in the river, the semi-floating eggs drift downstream. To survive and develop successfully, the eggs and larvae must reach inundated areas where the post larva and fry can obtain enough food and avoid their enemies. Let us talk about artificial and semi-artificial propagation. In that, first one is a band breeding. Band is a local term which means a dike or an embankment. However, with regard to fish breeding, bands are special types of perennial or seasonal ponds or impoundments, where riverine conditions are simulated during the monsoon season owing to the accumulation of rainwater from the catchment areas. The fertilized eggs are either allowed to hatch in the bund or are collected with the fine mesh nets. The bund spawning system might be considered primitive, but it has the advantage that it does not require hypophysation or specialized infrastructure. More up-to-date refinements of the bund spawning system combine traditional methods with the use of hypophysation. Nowadays, attention is paid to the proper selection of breeders, sex ratio and a definite stocking rate in a bund breeding. Some of the breeders from the selected lot, about 20 to 25%, are kept in a separate hapas according to sex. The females are injected with an initial dose of pituitary extract at 3 mg per kg body weight. After about 4 to 5 hours, the second dose of pituitary extract is administered to the females at 8 mg per kg. At the same time, males receive their single dose of 3 mg per kg. Then, the injected breeders are released back into the bund amidst an uninjected group of selected breeders. The inlet and outlet of the bund are open to allow a steady flow of water through the bund. The spawning activity of the injected breeders act as a stimulus to the uninjected ones to follow suit. It results in a mass spawning. Artificial propagation and fry production. The Indian carps are in every respect suitable also for artificial propagation. The brood stock can easily be raised in ponds if provided with the required space, healthy environment, natural and sometimes artificial feeds. Indian major carp brooders are normally raised in polyculture with other carps. Sufficient natural food can be provided by manuring the broodfish ponds. The selection of male and female fish with good potential for use as a broodfish is easy during or shortly before the normal spawning season. Females ripe for hypophysation have soft, bulging but elastic abdomens. The sexual opening and anus are swollen and pink or red colored. Males are more slender and usually ooze milt. The dorsal side of the pectoral fins are rough, as are the scales and head. Ideally, selected brooders should be injected and placed separately in water tanks immediately after measuring or estimating their body weight. Obtaining fertilized eggs. To induce spawning, a priming dose is administered between 16 hours and 18 hours. It contains 2 mg per kg of pituitary suspended in a physiological salt solution and glycerine. 6 hours later, females receive the decisive dose of 8 mg per kg body weight. Normally, freshly collected pituitary glands are used. Induced spawning can be expected 4 to 6 hours after the second injection. The optimal temperature is 24 degree centigrade. Acceptable range is 24 to 31 degree centigrade. The lack of disturbance at night and at dawn helps stimulate the natural spawning. Indian major carps can also be induced to spawn in breeding hapas. Box-shaped containers made of close meshed mosquito net cloth kept stretched by ropes at each of their 4 top and bottom corners tied to bamboo poles. After the first injection, selected brooders are placed in the hapas in groups comprising 1 or 2 females with 2 or 3 males. 2 equal doses of pituitary are administered, separated by an interval of 6 hours. The second dose is given at midnight and brooders then spawn in the hapas during the night. The next morning, hapas are checked to ascertain whether spawning has been successful. After removal of brooders, the fertilized eggs are removed and their numbers estimated by volume before transfer to a hatching hapa for incubation. Hatching hapas consist of 2 layers of netting, the inner layer being of mesh size large enough to allow the passage of a larva through it and the outer layer of smaller mesh to retain them. Thus, hatched larvae become separated from dead egg cells. Fishes of most species treated in the above way give behavioral indications that ovulation has finished inside them. Alone or in a company with males or other females, they make a typical spawning movements: head raised out of the water, spreading and twisting of the dorsal fin, swimming in circles, the so called swimming carousel, quivering, etc. For most species, the number of degree hours required between injections and stripping has been determined, but this is not yet known precisely for Indian major carps. It is also necessary to determine how long it takes after the proper spawning time for eggs to become overripe. This period varies between species, but is often less than 1 hour. Fertilized eggs are then transferred into incubation devices. Incubations of the eggs. Many different incubation techniques have been practiced with Indian major carps with varying success. Where no water current is available, incubation hapas are used, but in this many eggs frequently suffocate and die. Nowadays, some stations obtain eggs by induced natural spawning in circular or elliptical Chinese type spawning tanks, and the fertilized eggs are incubated in adjoining small round tanks receiving a continuous supply of water. Where flowing water is available, various types and sizes of glass jars are used. Instead, 2 alternative types of funnel shaped incubators are recommended. Soft incubators made of plastic sheeting with a sieve cloth collar designed for operation immersed in water. The usual volume of this is 40 to 50 liter, having a capacity of 1 to 1.5 lakhs eggs. Hard incubators made of fiberglass or other synthetic material. These are normally of 60 to 200 liter capacity and are equipped with filters to prevent the escape of eggs or larvae. In older models, the filters are fixed, whilst on newer ones they are changeable. The essential requirements of the developing eggs are suitable water temperature. For Indian carps, it is 27 degrees Celsius is optimal and ranges between 24 to 30 degrees Celsius. Continuous supply of oxygen to every egg in the incubator. Inflowing water should contain at least 5 to 6 milligram per liter dissolved oxygen. Removal of toxic metabolites like carbon dioxide, ammonia, etc., which is settled automatically by the continuous water flow. The water inflow should be regulated so that it does not cause mechanical damage to the eggs, which are especially sensitive in the early stages of development. Generally, turbid waters does not harm Indian carp eggs, ended in natural conditions, river spawning fish often spawn in very turbid water. The development time of Indian major carp eggs is temperature dependent and rather short, being similar to that of Chinese major carps. Next is rearing of viable larva until feeding starts. Separate special larval rearing devices such as happas are now rarely used, but sometimes newly hatched larvae are transferred to bigger, that is 200-liter funnel type systems. The environmental requirements of larvae are similar to those of developing eggs, except that they can tolerate a stronger water flows. The behavior of Indian major carp larvae is typical. After hatching, they first swim vertically, but after about a day, they lie down and are moved passively with the water current in the incubators. 1 or 2 days later, they begin to swim again and soon fill their swim bladders with air from the water surface. They are then ready to begin taking external food, and the post-larval stage of life begins. The first food offered to post-larvae in rearing funnel is usually either boiled egg, very finely ground, or micro-encapsulated egg diet. It is then advisable to stock the post-larvae as soon as possible into well-prepared nursery ponds. Nursing post-larva. When 3 to 4 days old, larvae are stocked into a well-prepared nursing pond. Fertilization of the nursery pond is commonly done with 10,000 to 15,000 kg of organic manure such as cow dung and 300 kg of lime per hectare. Generally, lime is applied before manuring. Inorganic fertilizer is normally not used. The nursery should have plenty of fish food organisms of smaller size before the larvae are stocked. Rearing in the nursery lasts for about 2 weeks. The rate of stocking in well prepared nursery pond with adequate fish food organisms can be as high as 10 million per hectare. Fry rearing. Rearing fry of about 20 millimeter length up to fingerling size, that is about 100 mm, is done in slightly bigger ponds of 0.1 hectare. Preparation of these ponds involves clearance of weed, eradication of aquatic insects and undesirable fish, and fertilization with organic manure. On growing to a market size, the Indian major carps are typical example of fish which can realize their potential for productivity and growth only in a polyculture. Let us talk about harvest. Final harvesting is done by seine net either during the summer months when the water levels falls, or after the monsoon, or when the market demand is highest. Now, let us talk about exotic carps. Mainly 3 exotic carps were also introduced for aquaculture, namely silver carp, that is Hypophthalmictus mollitrix, a grass carp, Ctenopharyngodon idalis, and common carp, that is Cyprinus carpio. Air breathing fishes. The culturable species of air breathing fishes are Channa striatus, that is also known as big or striped marrel or snakehead fish, Channa punctatus, that is spotted marrel, and Channa morulis, a giant marrel, Clarias batrachus, known as magur, Heteropneustes fossilis, known as singi, and Anabas testudineus, known as koi or climbing perch. Out of this, Channa striatus has highest demand in the market and is also commands a higher price. Seed collection. The seed of a marrel, magur and singi are collected from the natural resources in spite of success achieved in induced breeding. Marrels attain maturity in 2 years are known to breed throughout the year. The fry of 2 to 4 centimeter can be collected all around the year and from rain fed ditches and shallow water bodies with abundant weeds. The young ones emerging from the eggs move in shoals and their collection in large numbers is always easy. The fingerlings may not tend to move in shoals. Seed transport. The fry or fingerlings of air-breathing fishes can be transported without oxygen packing. Polythene drums or iron drums are used for transport of fry or fingerlings. The carrier must have enough space for their habitual surfacing to breathe atmospheric air. Pond management. Nurseries are about 10 to 15 square meter, having a water column of 50 centimeter. These are stocked with 0.2 to 1.5 million fry per hectare. Prior to stocking, manuring is done with a raw cattle dung at the rate of 500 kg per hectare alone. After nursery management, the fingerlings are to be transferred in stocking ponds. Stocking. Uniform size fingerlings are chosen for stocking. The fingerlings are disinfected with 2% potassium permanganate solutions for 5 minutes or dipped in a 200 ppm formalin solution for 50 seconds before stocking. Thereafter is a feeding. To maintain an abundant food supply for growing air breathers, the stocking pond must be rich in animal food source like frog tadpoles and trash fish. Growth and production. If the management practices are proper, giant and striped murrel can attain a growth of 1 to 2 kg per year and 0.75 kg per year respectively, whereas a spotted murrels grow to 160 grams in 8 months. Catfishes are known to grow slowly when compared to murrels. Magur and singi grow to 0.2 kg and 0.1 kg respectively. The conversion rate with recommended feed is approximately 2:1. Then there is a harvesting. Summer season is ideal for harvesting air breathing fishes from ponds. Now let us talk about a catfish. The catfish culture is limited and outdated and yet to be popularized among fish farmers. Catfish culture has a number of advantages over the carp, that is a greater survival in oxygen depleted waters, tolerance to the crowding, high stocking rates on artificial feeds, fewer intramuscular bones, tender flesh and a delicious taste. With regards to catfish, culture systems are yet to be established. In India, culture of magur catfish, that is a Clarias species, has been given a greater priority than the singi, that is Heteropneustes facilis. At present, most of the Indian fish farmers have directed their attention towards African catfish, Clarias gariepinus, due to the opportunity for short term profit, faster growth and a cheap mode of feeding. Now let us talk about cold water fishes. Culture of cold water fish in the Indian Himalayas has largely concentrated on the production of stocking material for rivers and streams and some lakes. Until recently, brown and rainbow trout and common carp highly dominated the fish cultured for fish seed production and to a much lesser extent for table fish production. Patliku hatchery in Himachal Pradesh is now producing 10 tons of table sized trout for the market and stocking material for streams. At trout farms in Kokarnag, belongs to Kashmir, and Katrine in Himachal Pradesh, high yielding strains of rainbow trout have been introduced. Success was achieved with artificial breeding of the indigenous cyprinid mahsir, that is Tor putitora and Tor tor, and Schizothoracinus, that is a Schizothorax richardsoni, Schizothorax niger, Schizothorax plagiostomus, Schizothorax plenifrons, Schizothorax carvifrons and Schizothorax ecosinus. Ornamental fish. The ornamental fish sector is a widespread and global component of international trade, fisheries, aquaculture and development. With the decline in the production from many capture fisheries, peoples are looking for sustainable harvest and culture of ornamental fishes. Ornamental fishery resources face a range of challenges, the need for their conservation and sustainable use. Now, let us discuss regarding the aquaculture of mollusk. Aquacultured shellfish include a various oyster, mussel and clam species. These bivalves are filter and or deposit feeders, which rely on ambient primary productions rather than inputs of fish or other feed. As such, shellfish aquaculture is generally perceived as benign or even beneficial. Depending on the spatial and local conditions, bivalve mollusks are either grown on the beach, on log lines or suspended from rafts and harvested by hand or by dredging. Abalone farming began in the late 1950s and early 1960s in Japan and China. Since the mid 1990s, this industry has become increasingly successful. Now, let us talk about the oyster farming. Oyster farming is an aquaculture or a mariculture practice in which oysters are raised for human consumption. Oysters farming most likely developed in tandem with pearl farming, a similar practice in which oysters are farmed for the purpose of developing pearls. Varieties of farmed oysters. Commonly farmed food oysters include the eastern oyster, Crassostrea virginica, the Pacific oyster, Crassostrea gigas, Bellon oyster, Ostrea edulis, the Sydney rock oyster, Crassostrea glomerata, and the southern mud oyster, Ostrea angasi. The cultivation. Oysters naturally grown in estuarine bodies of brackish water. When farmed, the temperature and salinity of the water are controlled or at least monitored so as to induce a spawning and fertilization as well as to speed the rate of maturation, which can take several years. Three methods of cultivations are commonly used. In each case, oysters are cultivated to the size of spot, the point at which they attach themselves to a substrate. The substrate is known as a culch. The loose part may be allowed to mature further to form a seed oysters with a small shells. In either case, spot or a seed stage, they are then set out to mature. The maturation technique is where the cultivation method's choice is made. In one method, the spot or a seed oysters are distributed over existing oyster beds and left to mature naturally. Such oysters will then be collected using the methods for fishing wild oysters such as dredging. In the second method, the spot or a seed may be put in racks, bags or cages, or they may be glued into vertical ropes which are held above the bottom. In the third method, the spot or a seed are placed in a culch within an artificial maturation tank. The maturation tank may be fed with water that has been especially prepared for the purpose of accelerating the growth rate of the oysters. Now, let us talk about a mussel culture. Culture of edible mollusk is now recognized as an effective way of enhancing food production, and sea mussels among all the known cultivable organisms. The green mussel Perna viridis and the brown mussel Perna indica are the two species occurring along the Indian coast. The green mussel enjoys a wider distribution along the east and west coast of India, whereas the brown mussel is restricted to the southwest coast of India. The mussel attain maturity at very small lengths. The green mussel attains maturity at 15.5 to 28 millimeter. Mussel spawns from the end of May till September. Larvae are free swimming for 15 to 20 days. The larvae attach to the substratum by means of the byssus. The metamorphosis takes place and the secretion begins. The young metamorphosed larvae that are known as a plantigrade is generally called a spot. The ability of the animal to regenerate the byssus is an advantage for transplanting the animal to new areas in mussel farming operation. The seed collection and seeding. The major part of the seed required for farming is collected on a ropes suspended from the rafts during the peak spawning period. The spot settled on ropes are allowed to grow to a length of 30 to 40 millimeter. These half-grown mussels are collected and transplanted to fresh ropes to avoid overcrowding and help further rapid growth and fattening. Mussel seeds are collected from the intertidal and submerged mussel beds after the peak spawning season, that is from September to November. The average seed size for farming is 15 to 25 mm and 60 gram seeds are required for seeding 1 meter length of the rope. The seeds are placed around the rope and securely warped with knitted cotton cloth. The seeded ropes are suspended from the rafts 0.5 to 1 meter apart, with the lower free end of the rope about 2 meter above the seabed. After the suspension of seeded ropes, the mussel culture farm needs only minimum attention to see that the rafts are in a good shape and the ropes with the growing mussels are hung properly. Now, let us talk about clam farming. A number of clam species belonging to the families Archidae, Veneridae, Corbiculidae, Solenidae, Talinidae and Donacidae occur in the coastal regions of India, and among these the most important species which are fished widely are Velorita cyprinoides, Porphia malabarica, Maritrix casta and Anadara granosa. However, there is no commercial culture of clam following a strict farming protocol, but a method of semi-culture whereby fisher stock seed clams occur in some region. Crustacean aquaculture. Commercial shrimp farming began in the 1970s and production grew steeply thereafter. Shrimp farming has changed from its traditional small scale form in the Southeast Asia into a global industry. Technological advances have led to ever higher densities per unit area. Virtually all farmed shrimps are pinnids, that is shrimps of the family Pinidae, and just two species of shrimp, the Pacific white shrimp and the giant tiger prawn, account for about 80% of all farmed shrimp. To sum up the topic, it can be said that a fishery in India is a very important economic activity and a flourishing sector with a varied resources and potentials. Now, fisheries together with agriculture had been recognized as an important sector. India is also an important country that produces fish through aquaculture in the world. India is home to more than 10% of the global fish diversity. Freshwater aquaculture contributes to over 95% of the total aquaculture production. The freshwater aquaculture comprises of the culture of carp fishes, culture of catfishes, into that air breathing and non-air breathing, culture of freshwater prawns, culture of pangasius and culture of tilapia. In addition, in brackish water sector, the aquaculture includes a culture of shrimp varieties, mainly the native giant tiger prawn, Peneus monodon. The three Indian major carps, namely Katla, Rohu and Mrigal, contribute the bulk of production to the extent of 70 to 75% of the total freshwater fish production. In addition, there is contribution from cold water fisheries, although insufficient for fish basket, is of high value and low volume category. Important food fishes of cold waters are mahasir and schizothoracids belonging to the indigenous species and trouts among the exotic varieties. Induced breeding of carps and catfishes, hatcheries for mass scale spawning, seed rearing and carp polyculture are some of the important technologies actually guided by the freshwater aquaculture development. The development of ornamental fish breeding and management has provided important livelihood options for marginal and landless farmers in certain localities. Mariculture in India, although limited to the farming of mussel and edible oysters undertaken in some coastal region of Kerala over the years, has successfully produced sea cage farming in recent years. So, that is all about the topic. Thank you.